thing I can do going through all these images is tell you all the things I can ask you.
Just take a picture.
No, I want to ask.
In my opinion,
Can you forward your recording?
So I'll wait for you guys to get all that stuff out. Let's see, go juice here. What we looked at, so it's going to start with epithelium, and it's going to connective tissue. Okay, come on in. This is probably going to, everybody that walks in, I'm going to have to start over. What was I saying?
Epithelium.
Starts with epithelium.
I have this in order, kind of like the way we got these things out. So epithelium first, simples first, then stratifieds, and then we go into the connective tissue, then we look at the muscle and the bone and everything. So I'll try to go through this as rapidly as possible. And I would really like you to all just sit down. These large cuboidal tubules are collecting ducts. That's what they are. So again, if you're taking down really copious notes here, you might want to label these things as collecting ducts. They are labeled in your lab atlas already. But in between here, these things are all, remember one of the rules of epithelium is all epithelium is lying over some kind of underlying connective tissue. Everything has some basement membrane to it, and what we're looking at here is the tubule embedded in its connective tissue. Now, this connective tissue happens to be just loaded with blood capillaries, and that's where you expect to find blood capillaries, is in the connective tissue. Connective tissue is loaded with vascularization, and all of these little dots here with the little oblique circles around it happens to be just loaded with blood capillaries, and that's where you expect to find blood capillaries, is in the connective tissue. Connective tissue is loaded with vascularization. And all of these little dots here with the little oblique circles around them are small, simple squamous lined capillaries. Okay? In the case of the kidney, these have a name. They're called the vasa recta. V-A-S-A-R-E-C-T-A, just in case you're interested in that.
What do you say was the black dots again?
Black dots are the nuclei of the squamous cells, and then the circles associated with them are, the way a capillary looks is like this. One squamous cell will form like a what? This or this or this. That one, that one, that one, that one, that one, and that one. What are they? They are ducts. How did you know they're ducts? Because they're lined with a cuboidal epithelium. What about that and that?
No.
Look carefully.
Blood vessels?
Those are blood vessels. These are not glands. These are lined with simple squamous epithelium. Those are blood vessels. These tiny ones, these big ones are all blood vessels. If you're hooking into the idea that you find the blood supply in the underlying connective tissue when you look at these kinds of tissues. If you haven't seen that yet, pay attention to that as we go through these various slides. Blood supply is always in the underlying connective tissue, especially when epithelium is concerned. Okay, this is the easiest one for, this and adipose tissue are the easiest ones for students to recognize. So what is this organ? It's a ureter. And is a ureter a blood vessel or a duct or what? How do you know? There you go. It's not surrounded by simple squamous epithelium, so therefore, by our rules, it's a duct. Now here again, we can see an epithelium with its underlying connective, and around that, what's this stuff here? Smooth muscle. And then here's some adipose tissue out here, a couple of blood vessels. So this slide would be good for transitional epithelium and smooth muscle. I am not ever going to put a pointer on the connective tissue and just ask you about that. But we can see a really good example of smooth muscle here. We can see a real good example of the epithelium. And if it's in the urinary system, this stratified cuboidal stuff we call transitional epithelium. Even though it was a mistaken assumption that caused it to be named that way, even though we now understand that it was a mistake, we retain the name. This is just a close-up of that ureter. So now, one of the things that you need to focus on, though, is we have ureter and bladder both for transition. The difference between the bladder and the ureter is the ureter, especially at low and medium power, is going to be a completely closed tubule, and the bladder is not. You're just going to see a portion of this epithelium just sort of moving across the field of image, but it's not going to be closed into a small tubule. Ureters are very tiny. They're just, no more than like a millimeter in their lumen diameter. They don't need to be very big, just little bits of fluid keep seeping through there. If you've ever, I've had the opportunity to see this once. I saw somebody pass a kidney stone, and those go from the kidney through the ureter into the bladder and then out the urethra. And the person was urinating through a tea strainer to catch it, to make sure it was out. That's what the doctor told us. Epitheliums. We only have two kinds of stratified epithelium, right? What are they? Stratified squamous, stratified cuboidal, and which transitional is one. I know your textbook makes reference of the fact that there is small examples of stratified columnar, but that's probably not true. I've never run across it in my experience, and I really have my doubts as to whether you could have a stratified columnar epithelium. You could have a stratified cuboidal epithelium with columnar basal cells, but by the time they get out to the free side, they're going to shrink down into some kind of cuboidal configuration. Now here we have, stratified squamous, which is what this is. You have two choices to identify it. One would, what this is, you have two choices to identify it. One would be the outer integument, and the other one would be the esophagus. How do you tell the difference? That's what you would look for. The outer esophagus has the stratum corneum over here. It's got that big, thick, dead layer of skin, and the esophagus doesn't have that. That's the only difference between those two stratified squamous epitheliums. Now, underneath this, you should have a pretty good idea of what all this stuff is. What do you think?
Smooth muscle.
Remember I told you that smooth muscle and one other kind of tissue were real hard to tell apart sometimes? Dense irregular connective tissue. The reason you know that this is not smooth muscle, columnar epithelium, but here you have this tubule that the fluid goes through, and it's this really thick stratified epithelium. Why do you suppose the ureter would, what advantage would this stratified tissue have in something like a ureter or a bladder? Why there? Why don't you have stratified cuboidal epithelium in sweat glands and in collecting ducts and things like that? They're all moving fluid through them. So why here would it be advantage to have a stratified thick epithelial tissue?
It's protected from the nitrogenous waste.
That's right, because nitrogenous waste, urea especially, is coming through here, and your kidney just went through a really big, although not perfect, effort to get rid of all the urea out of the blood, and the last thing you want it to do is diffuse back through the epithelium and the connective tissue into the blood, which it could easily do, except for that thick cellular barrier full of tight junctions and everything. And this prevents any kind of diffusion of urine back out into the body. We spend a lot of energy producing that stuff, and we don't want to just have it get reabsorbed back into the tissue. So you look at the structures in the urinary system that actually transport and house it, like the bladder, and you're going to see this rich, thick epithelium. Now, here in the esophagus, this is stratified squamous. And the reason that, well, I don't want to say the reason that this exists, but the advantage to having a tissue like this is this layer up here is a sort of an insensitive, it's not dead, but it's just a compressed kind of insensitive layer esophagus, but it's down a little lower. Now what about these things here? What do those look like? Look carefully, because it does kind of look like that's a stratified squamous
Blood
lining
vessels.
around there. But if you look more carefully, you can see that this is just the nucleus of a cuboidal cell that goes all the way out to
Okay.
there. And like, look here, you can see it really well. See the light lumen? This pink stuff is actually cells. So these are actually simple cuboidal cells. These are glands. If you see one moving along, it's probably a duct. If you see a cluster of them, that means it's some kind of a coil of glands. So what would you call these glands? Esophageal glands. Very good. So what I have here is stratified squamous epithelium, dense irregular connective tissue. I could ask you something like this. How many different types of fibers are described in areolar connective tissue? Described. 3. Because if you look up areolar connective tissue, like in your book, it will say that there are these big pink collagen fibers and the little black elastin fibers, and it also has reticular fibers. Okay. If you read that in the book, that would be a question I could legitimately ask you on the lecture test. But if you look in the slides and I ask you how many different types of fibers can you see there, the answer would be 2. Because we don't have any lymph nodes. Well, I tell a lie, I actually have one. I took the last one out before it got broken and kept it. But I'm not going to use it on the test because you guys didn't see it. So when you see reticular fibers here, there's nothing here. There's just reticular fibers and red blood cells, and this is the spleen. There's nothing there else that I could ask you. It's a loose connective tissue. It's called reticular. It makes up the spleen. This is what you're looking at. And all these cells are red blood cells. Now, I asked in the lab manual if you could see reticulocytes. You almost never do, but if you ever run across a spleen and you're looking through there and you see a cell with a nucleus in it, that is a reticulocyte, and all a reticulocyte is an immature red blood cell. Blood vessel right there. But all you can see here is red blood cells and reticular fibers. Okay? Any questions about this one? Good. Let's move on. Now come to the dense ones. Again, you'll find a lot of different descriptions of dense. One description used by Saladin, I believe, is that there's dense regular and dense irregular, and elastic tissue like this is just a certain kind of dense regular, which is the dorsal aorta comes out. And this black stuff in here is the elastic connective tissue inside the wall of the aorta. The aorta is a very big elastic artery. This is the cross section of the aorta, which we also don't have, so you don't have to worry about that.